Hello, can somebody confirm you can hear me?
We can hear you.
Great. Let me share my screen. Are there any questions before we begin? Should be seeing my screen now. So what we're going to do is just review for the final exam. The final will be due by 11:59 p.m. Wednesday, March 20th. That's next Wednesday. It'll be online. There will be heavy coverage of chapter 7, 13, 14, and lab module 9. So to begin this review, I'm going to start with the chapters we have heavy coverage of, and then I will talk a little bit about the chapters that have a few more questions in them than the other chapters. So most of the other chapters, other than chapters 7, 13, 14, and module 9, will only have like 2 or 3 questions in them. There's an exception with chapter 8. It has a little bit more questions. And you can see I budgeted 3 days for discussing chapter 8. It's a little bit longer chapter and we went into it in a little more greater detail. So I'll spend a little more time on chapter 8. And then the other chapter I want to spend some more time on is chapter 1. Chapter 1 is a chapter that introduces a whole bunch of concepts. And then later on, we talk about those concepts. We first ran into endospores and Koch's postulates in chapter 1. And then we talked about both of those topics at 2 other times in the class. And so I want to talk about chapter 1 in a little more detail because there's going to be more questions on chapter 1. And one of the reasons is, of course, this chapter covers a whole lot of material very briefly. And then it goes into the history of microbiology. And I have a number of questions about the history of microbiology. Any question about any of that? All right, so let's begin. I'm just going to begin at the back. I'll start with lab 9. And come on. Oh, geez, I lost control here. There it goes. Surprisingly, lab 8, I don't really have many questions on it in the quizzes or in the final. And the reason for that is lab 8 was added after the online class was made. When COVID-19 came around, everybody went online, and lab 8 was added to microbiology after the class was developed. And so I just never went back and added many questions about lab 8. So lab 8, you kind of get, I don't know, free show for it. But lab 9, there are some questions on the final from lab 9. So let's talk about lab 9 first. And I don't think I have this in the right place. So let me open up another one. I have it for discussing the the lecture modules, not the labs. So lab 9 was identification of gram-negative bacilli. Particularly, we looked at the family Enterobacteriaceae and the family Pseudomonaceae. Both of these families look the same. They're short rods. They're both gram-negative. But they have some differences, like the Enterobacteriaceae. All of them can ferment the sugar glucose. And the Pseudomonaceae, none of them can ferment the sugar glucose. There are some other differences too, and that is the Enterobacteriaceae are all oxidase negative, and the Pseudomonaceae are all oxidase positive. So in lab 9, you should talk about, be able to explain the Einvik reactions, meaning be able to explain the indole test, the methyl red test, the Voges Prescure test, and the Simmons citrate test. And something that's easy for the indole. Hello, let me stop the share here. Somebody had their microphone on, but they've turned it off now. So something that's easy with the indole test, the methyl red test, and the Voges Prescure test, if the test is positive, meaning the result, it'll be red in color, all three of them. There are some differences, like the indole test is only at the top, and the methyl red is throughout the entire tube. But other than that, it's red if it's positive, it's yellow if it's negative. For the Simmons citrate test, it is blue if it is positive, and it is green, no change, if it is negative. Oh, come on, lost that. And then we want to talk about the carbohydrate fermentation test. We've talked about that before. The motility test, the hydrogen sulfide production test, and I'll go through these with the pictures. And then more difficult to explain how to interpret the TSI, triple sugar iron test, the SEMS test, and then the growth on McConkey's agar and hectone enteric agar. And then lastly, use biochemical analysis to study case studies. There will be a few case studies. Most of them will not be in the multiple choice test because that test is timed. And so if I give you a case study in a multiple test time portion, it'll be a fairly simple case study where you should be able to look at one or two tests and then quickly be able to identify the species. More likely, I would give you a case study in a question which would be a non-test exam. Let me take a look at the exam here. I think I've got both types of questions, but oh, come on, boy, this is really slow today. So I think you have to take both finals, both a timed one and an untimed one. The untimed one, yeah, final A, it's only worth 9 points, and that probably means it's only got 3 questions. So at most, there probably will be one case study. And then final B, if they're, like I said, this is a time exam. And so if I have them in there, they'd be fairly simple. All right, let me go back to where it was. Some of the family of Enterobacteriaceae do have frank human pathogens like the genera Salmonella, Shigella, and Escherichia, although it's only certain variants of Escherichia like E. coli, Suravar 015787. Pseudomonas aeruginosa is the main Pseudomonas, which is a human pathogen, but it's very lousy at getting established on a healthy, normal individual. It's more likely to be an opportunistic pathogen, particularly of immunosuppressed individuals, burn patients, and trauma patients. And the burn and the trauma either damage the mucous membrane or the skin, allowing Pseudomonas to get established, or the individual is immunosuppressed and then it can get established. It is very lousy at getting through intact skin and intact mucous membranes. All right, so let's talk about some of the tests. I already talked about the indole, the methyl red, and the Voges per Scure test. All of them are looking at products, and if it's positive, the test will be red in color. Citrate utilization, looking to see if the organism can grow with citrate being the only carbon source. And if it can, it'll turn blue, and you should be able to see some growth in the tube. If it's green and no growth in the tube, then it's negative for the Simmons citrate tube test. For the PR carbohydrate metabolism test, this will look at the fermentation of one sugar. There's only one sugar in the tube. And if the bacteria can ferment that sugar, it will change the color from red or pink to yellow, because fermentation will create acid products from the sugar, changing the pH to making it yellow. You can have two positive reactions, A, just yellow, and then yellow with, or acid with the production of gas. So we go AG. Both of those are positive, and this is negative. Any question about the fermentation test? It may be called glucose fermentation, sucrose fermentation after the sugar, or it could be carbohydrate fermentation test, just talking about it in general. If not, let's move on to the motility test. The motility test uses a semi-solid agar, meaning very low agar concentration, where the bacteria can swim through the agar. You just stab inoculate, and if the growth is only in the stab, the bacteria is non-motile. If it's highly motile, you'll get the bacteria to go everywhere, and the tube will look cloudy from the bacteria growing everywhere. If it's slightly motile, the bacteria will be flaring out from the stab mark. So instead of being only in the stab, it'll flare out sort of like a flame from a candle flaring. All right, any question about the motility test? If the bacteria is motile, it has a flagella or flagellum if there's only one. The motility test may be done on more than one media. You can test it in the Sims tube. You can do it in the motility test media, which is what we're showing here. Hydrogen sulfide production is looking for the production of hydrogen sulfide. If hydrogen sulfide is produced, the tube will turn black. You can test this with the Sims tube, the triple sugar iron or TSI test, or on the hectone enteric agar plates. So there's a Sims tube, and the bottom portion is black. Hydrogen sulfide is always made anaerobically, which is why it's usually in the bottom of the tube. All right, any question about hydrogen sulfide production test? If not, let's talk about the triple sugar iron test. This test is a complicated test looking at several different things going on at the same time. First, you stab inoculate to get growth at least partially anaerobically, if not completely anaerobically. You then grow the culture on the slant to look at the growth aerobically, And then you compare the growth aerobically to the growth anaerobically. You also look at the production of hydrogen sulfide. It'll be black in the bottom of the tube. Hydrogen sulfide is made. And then we're looking at the fermentation of the sugar, both in the slant and in the butt. Oh, and then one other thing, you're looking at the production of gas if fermentation occurs. I'm on this tube here, so let's just quickly talk about gas. If you have gas, you'll get a bubble, usually in the bottom of the tube, which will lift the auger. You may also just get cracking of the auger. Those are two signs of gas production. No gas production, there's none of that. A negative tube will be pink or red throughout the tube, no change. And that can be abbreviated K over K. K stands for alkaline reaction. It could also be negative or negative. If you get fermentation in the slant and in the butt, we go A for the slant for acid fermentation in the slant over acid in the butt. If that happens, glucose, and then at least one other sugar is being fermented, and that could be lactose or sucrose, or maybe both. We don't know, except that it's glucose and one other sugar. That's all we know. We know it's always glucose because glucose is the base sugar. The other sugars are actually converted into glucose and then sent down aerobic respiration in the normal way using glucose. So glucose would always be fermented if one of the others is fermented. Any question about that? Okay, the tube has glucose at much lower concentration, 1/10 the concentration of the lactose and the sucrose. And so if the glucose is the only sugar that is fermented, it'll be red in the slant and yellow in the butt. The reason for the difference is in the slant, the bacteria can grow quicker than in the butt, and all of the sugar can be used up. And rather than the bacteria dying, they start utilizing the protein in the media. And if they utilize the protein, they make ammonia. Ammonia is a base, and the base that's being made will neutralize the color, which if we looked at this earlier, it would have been yellow. But at the time span of the test, the ammonia starts getting produced, and that will switch the slant from yellow to a pinkish color. The butt, on the other hand, the bacteria grow slower, they ferment the sugar slower, and so the butt will look yellow. So if glucose is the only sugar being fermented, the butt will be yellow, the slant will be red or pink. Any question about that? All right, in the case of hydrogen sulfide being produced, the black will hide the yellow, but you can always know that hydrogen sulfide is only being produced when the sugar is fermented. So if it's black, it's always yellow. In this case, the slant is not yellow because only glucose is being fermented. Any question about that? And I know the triple sugar iron tube is fairly complex. Just do your best and I guess study this figure. Sulfide indole motility is the Sims test, and it's looking at 3 tests at one time. First, we're looking for hydrogen sulfide production. And if that is produced, it'll turn black in the tube. So tube 1, hydrogen sulfide is produced. The indole for the I is for the indole test. And what you do is you add Kovacs reagent to the top of the tube. And if indole is produced, it'll turn pinkish or red. And so these tubes are indole positive, these two are indole negative. And it stays yellow if it's indole negative, or yellowish. That's sort of a whitish yellow. That one's a light yellow. And then the M, we're doing the motility test. You stab inoculate the bacteria. If the bacteria only grow in the stab, as tube 2 or tube 3, that one's hard to see, or tube 5, then the bacteria is non-motile. If, on the other hand, the bacteria flares out from the stab or goes everywhere, then the bacteria is motile. And this one you can see it's flaring right here. And so the bacteria is motile on tube 4. Any questions about the SIMS tube? If not, let's talk about McConkey's auger, and then I'll talk about HE auger. Although both of them are looking at the growth of gram negatives, these plates select for gram negatives, select against gram positives. And so if there is growth on the plate, either one, then they are likely to be gram negatives. Both plates are also looking for the fermentation of the sugar lactose. On McConkey's, if the sugar lactose is fermented, the cellular colonies or the growth will appear pink, and then the media around the growth will also appear pink. It's from the fermentation of sugar changing the pH of the media. In this case, it becomes pink when it is acidic. In a non-fermenting or non-lactose fermenting bacteria, the bacteria will be their normal color. In this case, it's white or off-white, but it would depend on what bacteria we're streaking here, because the bacteria is its normal color. Any question about any of that? All right, for the HE plate, we are also looking at lactose fermentation, and if the bacteria ferment the sugar lactose, they'll be salmon colored. That is a yellow-orange color. If the bacteria do not ferment the sugar lactose, they will be their normal color, which this case is white or off-white. Any question about that? Now, the HE plate can also look at the hydrogen sulfide being produced. And if hydrogen sulfide is produced, the colonies will be black. And they will be black either with the fermentation of the sugar, in which case it'll be very hard to see the salmon color. But what you can do is, let me blow this way up. Come on. There we go. You can look at the media around the colony, like this orange colony. There's a reddish orange color around the colony. You can look there because the colony will be black. You won't see the salmon color. Any question about any of that? All right. And then you should know a little bit about the case studies because there could be a case study in the exam. Any question about Lab 9? If not, let's begin with the last chapter we studied, chapter 7. So obviously you should know the terms, be able to discuss the factors that influence microbial death rates, be able to discuss the three main ways in which antimicrobial agents kill or inhibit cells. And those three ways are, one, they damage proteins or what's the word? Drawing a blank here. cause the protein to unfold, which has a name, and I'm losing the name there. And that's the first way. The second way is to damage the membranes, especially the cell membrane. You damage the cell membrane, the cytoplasm will leak out. That'll kill the cell. And the third way is to damage DNA or nucleic acids. You do that, you cause mutations, and you get enough mutations, it'll kill the cell. Coagulate the protein? That's not the word I was looking for. Still losing it on the protein. All right, and then understand the various types of disinfectants and how they work. Just kind of know a little bit about the disinfectants I discussed in today's lecture. And then understand the relative resistance of the major microbial groups to antimicrobial agents. Any question about any of that? So you should know that microbial death rates are influenced by the number of microbes. The more microbes you have, the longer the treatment needs to be in order to kill all the microbes. And then the time of exposure is obviously important for affecting the microbial death rate. Generally, longer is better. Environmental influences can affect the microbial death rate, and that's mainly that organic matter may influence or inactivate the antimicrobial agent. And then microbial characteristics can affect the microbial death rate. As I mentioned, the mycobacterium have mycolic acid in their cell wall, and that gives these cells, I don't know, not totally invincibility, but partial invincibility to any agent which is water soluble. It keeps the water soluble agents out of the mycobacterium. You know the terms. Denature, that's the word I was looking for. Denature the protein, damage the intercellular proteins. You should know the methods for controlling microbial growth. You should know that wet heat is more effective because it transfers the heat quicker and better. And we all know that. You can't put your hand through steam, but you can put your hand in a oven that you just baked something in. You should know about pasteurization, the two methods for pasteurizing something like milk. You should know about filtration to remove the microbes, and it actually sterilizes the whatever you filter. Low temperature inhibits microbial growth. It does not totally prevent microbial growth, and the psychotrophs can grow at refrigeration temperature. So you should know that. High pressure can control microbial growth. Desiccation can control microbial growth. And I think we all know that molasses and honey can stay at room temperature for years and it doesn't go bad. And like I said, I proved it. My mother's molasses that we figured was at least 20 years old. I didn't eat any of the candy, but I'm a hypoglycemic. But my father did, and he was fine. He enjoyed it. You should know about radiation to control microbial growth. There's two types of radiation: ionizing radiation, which causes ions, and then UV radiation, which causes thymine dimers, both of which mutate the DNA and can kill the cells. You should know about the chemical agents for controlling microbial agents. You should know about phenol. You should know about halogens, especially bleach and iodine. You should know about alcohols, heavy metals. We didn't really talk much about chemical food preservatives, antibiotics, surface active agents, aldehydes. I did talk a little bit about gaseous chemo sterilizers, where you can apply a gaseous agent to the microbe, and it does kill everything, including endospores, but it does take a long time exposure to kill endospores. And the trouble with the gaseous agents is they're highly toxic to us. The peroxygens are an oxidizing agent, and they are actually one of the few agents which can kill endospores. All right, I think that's mostly it. You should know about the microbial characteristics for killing various major microbial groups. Any questions about chapter 7? If not, let's move on to chapter 13. Where am I? Viruses, viroids, and prions. Of course, you should know the terms, understand the characteristics of virus structure and viral classification, and understand about viral multiplication. The prokaryotes, meaning bacterial, let's only say bacteriophages, the viruses which infect kingdom bacteria or domain bacteria, only reproduce in two ways, either the lytic life cycle or the lysogenic life cycle. Now, animal cells reproduce in a few more ways. They mostly reproduce either in the lytic life cycle or in the budding life cycle. However, they do have two other life cycles. They have the latent life cycle where the virus does not reproduce. The virus is just in stasis in the latent life cycle. It doesn't cause disease. It's just hanging out, waiting for the virus to reactivate. And the other, we're not really going to talk about a persistent viral infection, which is similar to the budding life cycle, except it doesn't bud. You should know that the budding viruses always have an envelope. Any question about that? All right. And then lastly, understand about prions and viroids. You should know that viruses display no or little metabolic activity outside of their host cell. They rely on their host cell for many molecules made by the host cell, like ATP. There's a host range of a virus, and that's the range of cells a virus can infect. Most viruses only infect a few cells of one or a few species. Like HIV only infects like the human T cells and not human, the helper T cells, as well as the macrophages and brain cells. Does not infect many of our cells like skin cells or muscle cells. That host range is dependent on the molecule of the virus, the adhesions and the ligands that bind to the molecule of the host cell, and we call those molecules receptors. You should know about viral size. You should know viruses have a nucleic acid molecule inside them. It's either DNA or RNA, never both. And if it's, doesn't matter, DNA or RNA, it can be double-stranded or single-stranded. With an RNA molecule, it's important to discuss whether it's positive single strand or negative single strand. If it is positive single strand, the ribosome can read it. The ribosome cannot read the genome of a negative single strand RNA virus. You know about the capsid and the envelope on some viruses. The general viral morphology, their shape, come in 3 types: helical viruses, polyhedral, and then complex, where the virus could be anything. You know T4 is the most studied virus of all viruses. You know about viral taxonomy. We don't have much above the family. We do have whatever is immediately above a family, which is either a class or an order. I think it's an order. And then we group the viruses into orders, families, genus, and then virus species, and then subspecies. You should know that a subspecies is just designated by a number, and it's extremely important for a virus. The best example is with the polio virus. Subspecies 1 causes the polio. Subspecies 2 we use as a vaccine to prevent getting polio. So the Sabin vaccine has a live virus, which we use as a vaccine, and it doesn't give the polio. It prevents people from getting the polio disease. All right, talked about the lytic life cycle. Let me talk about one consequence of the lysogenic life cycle, and that is a cell that gets a lysogenic virus in it, gets new DNA added to the cell, because that lysogenic virus will integrate into the host cell's chromosome and then can be expressed. So a cell can get a new phenotype because it's getting new DNA, the DNA from the virus. The best example is Corynebacterium diphtherium only produces the diphtherium toxin when the bacteria is infected with the lysogenic virus. If the cell does not have a lysogenic virus, it does not produce the Corynebacterium diphtherium toxin. And the reason is that the toxin is encoded on the DNA of the virus. Any question about any of that? All right, so let's talk about animal viruses. Animal viruses replicate and divide and all that. Not divide, replicate, the same thing. They have a life cycle similar to bacteria, but there are some differences. First, there are some differences infecting the cell, and that's because it's much easier to get into a cell membrane than through a cell wall. Animal viruses infect animal cells by either having their envelope fused to the cell membrane or they're brought into the cell by endocytosis. The other main difference of animal viral multiplication is prokaryote cells, bacteria viruses, can only replicate in the cytoplasm because that's all the host cell has. But animal viruses may replicate in the cytoplasm, but they may replicate in the nucleus. And it just depends on the species. As a rule of thumb, if the virus is a DNA animal virus, it will replicate in the nucleus. And I don't know of any exceptions. With the RNA animal viruses, when they infect a cell, they replicate in the cytoplasm. And that's a little oversimplified simplification of the problem, but there is one exception and you should know it, and that is the retroviruses do not replicate in the cytoplasm. They replicate in the nucleus, even though they are an RNA genome. Any question about any of that? Already talked about that. How a envelope virus obtains its envelope is it actually buds out of a membrane of the cell and then rips off a portion of that membrane to form the envelope of the virus, and then the membrane will reform. It'll just reanneal. And that membrane could be the cell membrane, which is sort of shown here, could be the nuclear membrane, or it could be the membrane of the endoplasmic reticulum. It just depends on the viral species. Any question about any of that? No, envelope viruses are always released by budding. That's how the virus gets its envelope. The non-envelope viruses are not released by budding. They're mostly released by lytic litosis or lysis of the host cell. There is one exception, and that's the persistent infective virus, which it'll be released by a cell vesicle, and you don't really need to know about that category of virus. You should know about budding viruses and then lytic animal viruses. And then you should know there's such a thing as a latent animal virus, where the virus leaves the budding life cycle, does not replicate, and it enters the latent life cycle, where the virus is just not really being metabolically active. All it is being in stasis, waiting for the virus to reactivate. All right, so we talked about the DNA family of viruses. Do not memorize this slide here. I'm trying to, let's see, it's here. Get this to blow up. You should know Poxviridae contains the smallpox virus, and that's probably all you need to know about Poxviridae. The herpes viridae contain the herpes viruses, and that's a large number of viruses that tend to infect humans, like herpes simplex type 1, herpes simplex type 2, chickenpox, or later on shingles is caused by a herpes virus, cytomegalovirus, Epstein-Barr virus, which can cause mononucleosis. We're not going to talk about any of these families here. You should know that Adenoviridae is a DNA virus that is the third family and least likely family to cause a human cold, but it can cause a human cold. We're not going to talk about any of the other families. Oh, come on. Well, you should understand about viral multiplication. You should know there are more families of RNA families of viruses that can infect humans. So there's 15 known families that cause disease in humans. All right, you should know that retroviruses have the enzyme reverse transcriptase, which is very important because reverse transcriptase can make single-stranded DNA from RNA. And then the reverse transcriptase can convert the single-strand DNA into double-stranded DNA. The double-stranded DNA then goes to the nucleus if it isn't there to begin with, and it can bind with the host DNA, which may cause cancer later on. We'll talk about that in a little bit. Any question about any of that? The family Retroviridae comes from RE reversed and then TR for transcriptase. So that's where the name comes from. All right, you should know a little bit about coronaviruses, and they were long known to humans as viruses which infect children, particularly small children. And they're also known for COVID-19, causing COVID-19, or SARS, or MERS. Any question about any of that? All three, SARS, MERS, and COVID-19, can be lethal when it's has infected a susceptible host. COVID-19 was likely to be a recombination between two bat coronaviruses and one pangolin coronavirus. And apparently with COVID-19, recombination can happen. It actually happened once when we were studying the virus, and that is the variant Omicron was a combination of COVID-19 and a coronavirus that causes the human cold. And that's why Omicron was less lethal than any virus up to that date, whenever Omicron came around, which I think that's like 2 years ago now, but it might be longer. And this they know from the sequencing of the coronavirus. You should know the signs and symptoms of coronavirus. You should know that some viruses cause cancer. So about 10 to 15% of human cancer is caused by a virus. The best well-known human papillomaviruses can cause cervical cancer and skin cancer. And then hepatitis B and hepatitis C virus can cause liver cancer. Any question about any of that? All right, showing you the course of an infection, depending on the different types. An acute infection, the virus gets inside a host, begins incubating, and then very quickly the viral numbers increase, and then they decrease very quickly. So the signs and symptoms are actually short-lived from when you first have the signs and symptoms to when you no longer have the signs and symptoms. And for most human colds, it's about a week, maybe less. I'd have to look that up, but it's around that time. That differs the acute infection from a persistent infection. A persistent infection is where the patient gets infected and then it slowly grows over time. In the worst case scenario, we get a bloom of the virus, and then the virus kills the patient while the graph ends right there. You could, however, get a persistent viral infection, which just never goes away, but it never gets worse. And so it climbs, and then it just plateaus. Any question about persistent viral infection or an acute infection? If not, let's talk about a latent infection. A latent infection is when the patient gets shingles and previously the patient only had chickenpox. So a latent infection is one where the individual previously was infected and they probably built up an immune system that kept the virus away until the immune system weakens, and then the virus can come in and infect a similar individual, like a similar stock of grain. Because it's only been 10 years since we separated the bacteria from its host. So it hasn't evolved enough to not recognize its host cell. All right, any questions about any of that? I think we're done with this lesson, but let me look. All right, you should know what prions are, a proteinaceous infectious protein. You should know how prions replicate. and that is the prion protein simply comes up to the normal cellular protein, the PRP superscript C protein, and then causes the PRP superscript C to unfold and take on the folding pattern of the scrapies, cell. So the PRP C, the normal cellular protein, and the scrapies protein, the PRP superscript SC, has the same amino acid sequence. The only difference is the folding pattern of the protein. All right, you probably don't need to know about viruses. You should know that viroids are a naked RNA molecule, and they are infectious. When they get in a cell, they can cause disease, but there are no viroids known to infect and cause disease in animals. We only know of viroids in the plant kingdom. Any question about any of that? All right, if not, that's it for chapter 7. Let's move on to chapter 14. With this chapter, it's important. I got the wrong thing there. It's important in this chapter to know the terms. If you know the terms, you will know. Oh, fudge. I shut that down. If you know the terms, you'll know 50% of the lesson. Finally, I got it. Understand in this chapter about the relationship of the normal microbiota and its host. And you should understand where the normal microbiota can grow in the host and where it cannot. Understand the stages of disease development. And then lastly, and most importantly, understand about nosocomial infections and how to control them. The reason why that's so important is you guys are mostly going to go in the healthcare industry. So you should know what nosocomial infections are and how to prevent them. Terms, we're going to skip the terms. You need to know the terms. So the normal microbiota is found on the skin. All of the mucous membranes open to the outside. All of the gastrointestinal tract, the lower urinary and reproductive systems, like the urethra, has a normal microbiota. At least in women, the bladder also has a normal microbiota. Anything else? We do not normally find the normal microbiota internal to the body, such as in the blood, the cerebrospinal fluid, muscle, or nerve tissue. If it's there, that means we have an infection. The normal microbiota generally colonizes the host for almost their entire lifespan. So after a baby is born, they get their normal microbiota, largely from touching their parent or the parents touching them. And the normal microbiota will continue to be with the baby for most of the baby's lifespan. That differs from a transient microbiota, which may be present for days, weeks, or months. And the best example I know is when I had Staphylococcus aureus on my skin after hugging my aunt who had a Staphylococcus aureus skin infection. And fortunately, the next time I sampled my skin and all other times afterwards, which I probably did it for at least 3, maybe 5 years later, and I did it every term. I didn't have any Staphylococcus aureus any other time. So that was a transient microbiota. Any question about any of that? should know there's more normal microbiota than human cells, and that the microbiota are found in some unusual places like the bladder of a woman, as well as the breast tissue making milk of a woman. You should know the relationship of the normal microbiota to its host is either commensalism, meaning commensalistic symbiosis, or mutualistic symbiosis. Symbiosis just means the two organisms live together. Mutualism, where both organisms benefit from the arrangement, like E. coli gets a home, E. coli gets food from us, and then E. coli helps us digest our food. Commensalism is where one organism benefits, the other is unaffected. The best example I know is there's a nematode in her eye, and it lives in her eye. It does not do damage to her eye. So it is a commensalistic relationship. Any question about any of that? Oh, the benefit for the organism is it gets a home. We don't really feed it. So the only benefit it gets is it gets a home. All right, you should know about opportunistic microorganisms. That's when a normal microbiota causes an infection. And this does not normally happen in a healthy individual, but it can happen under certain circumstances, such as when the patient is taking immunosuppressive drugs, or when some predisposing disease has damaged the body, like the skin or the mucous membrane, and then the normal microbiota can start growing in that region. And that would be an opportunistic pathogen. Like E. coli, if it's found in the gut, everything is fine. However, if E. coli is found out of the gut, like in the inner ear or in the lungs, it would cause disease. Any question about any of that? All right, you should know about Koch's postulate. I'm certain that there will be at least one question on Koch's postulate on the final, because we ran into Koch's postulate 3 different times. It is the step he came up with to prove that a given microbe causes a given disease. And Dr. Koch first showed that Bacillus anthracis, that bacterium, causes the disease anthrax. That was the very first time we had a microbe that was shown to cause disease. You should know about the exceptions to Koch's postulate, where when we can't use Koch's postulate, we fall back on very large epidemiological data, or a very large epidemiological study, which follows a patient over time. And that's how we've shown that HIV causes the disease AIDS. All right, you should know about the terms of the occurrence of disease. You should know about latent disease and herd immunity. You should know about the predisposing factors that make a person more likely to come down with the disease, like the shortness of the urethra in females, more likely to give them a urinary tract infection. Inherited traits like sickle cell gene make it more likely that a sickle cell patient will come down with another disease, an infectious disease. The climate and weather, fatigue, both of these can affect the occurrence of disease. Age, generally, the older you are, the more susceptible you will be to getting a disease. Lifestyle and then chemotherapy both can affect the individual lifestyle. Like smokers are more likely to get a respiratory disease because the smoking damages the mucous membranes of the nose, throat, and upper respiratory tract. Chemotherapy weakens the host immune system, making them more likely to come down with the disease. You should know about the development of disease. All diseases follow these 5 periods. There's an incubation period, a prodromal period, where the signs and symptoms are mild, a period of illness where the signs and symptoms are at their worst, the period of decline where the signs and symptoms start getting better, and then the period of convalescence where the number of microbes have decreased in the individual below a threshold number so that the individual has no further signs or symptoms of disease. However, they still have the disease carrying organism in them. You'll never get rid of the organisms until you're in the, I guess you'd call it, it's not a period, but it's when you're cured after the period of convalescence. You know about reservoirs of infection and transmission of disease. And then lastly, you should know about nosocomial infections and how to control them. Any question about any of that? All right, that's it for chapter 14. Let's quickly move on to chapter 13. To know the terms of this chapter, understand the characteristics of viral structure, viral classification, understand about viral multiplication, and understand about prions and viroids. Have I discussed this chapter? Anyone?
Yes.
I have discussed this chapter? Sorry, let's move on to chapter 1 then. No, let me briefly talk about chapter 8. Be able to describe DNA replication, RNA transcription, and protein translation. Also be able to understand the different types of a mutation. And then lastly, of course, know the terms. I think in the beginning, it's all terms. You should know about the central dogma in biology, and that is information is stored in the DNA. That information is transcribed into RNA, and then that information in RNA is translated into the information in the protein. You should know that we can talk about the flow of genetic information. There's the flow of genetic information within a cell, which I just mentioned. Then there's this flow of information between generations of cells, where a parent cell sends that information to the daughter cells. There's also the flow of information between cells of the same generation, where DNA leaves one cell, goes in the environment, and then is taken up by a recombinant cell. You know that DNA replication is semi-conservative, meaning one strand, the old strand, will be used as a template to make the new strand. And semi-conservative means that the daughter strand will have, the daughter double strand of DNA will have one single strand, which is the old template, and then one newly replicated strand of DNA. I told you should know about these. Okay, you should know about bacterial gene expression. Cells have ways to control their gene expression. We're only going to talk about one way, and that's their inducible operon. An operon is a super gene. It does include the gene of interest. Like if we're talking about the enzyme lactase, that would be the Z gene in the lac operon. But there are other structural genes in the lac operon, and then there are other controlling regions of DNA around the lac operon. And that would be mostly the promoter, the operator, and the terminator. Terminator is not shown here because it's so small, but it's at the end of the gene. In the case of the lac operon, the lac structural gene is normally turned off. It's turned off by a repressor gene, which makes a repressor protein. That repressor protein binds to the operator. And then when the RNA polymerase binds to the polymerase, it cannot, or will try to move down the DNA, but once it runs into that repressor protein, the RNA polymerase cannot go further, and that prevents the lactase gene from being transcribed. Any question about any of that? Well, you need to have some way to turn on the lac gene, and you want to do this whenever the sugar lactose is around, because the bacteria would want to metabolize the sugar lactose. That's especially true if there are not other sugars around. How you turn on the lactose operon is we have an inducer, and it's allolactose. That's just an isomer of lactose. So if ever lactose is around, allolactose is around. And I used to know this. I think lactose is the L form, left-handed, and allolactose, I think, is the right form. But don't quote me on that. It's not terribly important. I'm not going to look it up. How the inducer turns on the lac operon is the inducer, allolactose, binds to the repressor protein and inactivates it. If the repressor protein is inactivated, it cannot bind to the operator, and that will allow expression for the Z gene. Now, what happens to the repressor protein that's bound to the operator is every once in a while it falls off. And if there's lots of inducer around, it'll bind to the repressor protein and inactivate it, so it can't bind to the operator any further. And that turns on the gene. Then the polymerase can bind to the P and then move down the DNA, transcribing the Z gene into messenger RNA. What do you notice is different about this messenger RNA from all other messenger RNAs we've talked about? All right, you should know about that difference. And mainly that in the case of an operon in bacteria, you can have more than one structural gene in that messenger RNA. One messenger RNA is actually going to code for 3 different proteins. Beta-galactosidase is another name for the enzyme lactase. Permease is the transporter protein that brings the sugar lactose into the cell. And then transacetylase is an enzyme that helps convert the sugar beta-galactose into the sugar lactose. Any question about any of that? All right, you should know about mutations. What's a spontaneous mutation? And there's 3 types. A base substitution has a silent mutation, a missense mutation, a nonsense mutation. You should know about frameshift mutation and macromutations. When you're learning about macromutations, please pay attention to this lesson because macromutations are not discussed in your textbook. All right, you should know about recombination and crossing over. You should know about vertical gene transfer. Cells, not vertical gene transfer, horizontal gene transfer. Cells can transfer genes among cells of the same generation between a donor cell and a recipient cell. Cells can do this by one of three ways: transformation, conjugation, or transduction. Transformation involves DNA from outside the cell being incorporated into the cell, and then a portion of that DNA is incorporated into the chromosome. And once that happens, that gene can be expressed. That may change the phenotype of this cell. Conjugation happens in two ways, either by a sex pilus if it's a gram-negative cell, or a mating bridge if it's a gram-positive cell. We're going to talk further about the gram-negative conjugation. Why isn't that moving? Oh, I'm in the wrong place. Conjugation encodes for the F plasmid encodes for the creation of a sex pilus. The sex pilus actually goes out into an F minus cell and skewers it, and then winches the cells together. They do not have to be this close. They can be this far away from each other, which is, I don't know, like 5 widths of the big cell, the fat cell. Anyways, the F plasmid can replicate and then transfer into the F minus cell. That will change the F minus cell to make it an F plus cell. Any question about any of that? And then transduction uses a virus to move DNA from one cell, this cell or that cell, to this cell. It rarely happens, but all three of them, sexual conjugation, transformation, and oh, I lost one. Transduction, conjugation, and transformation can all move DNA from a pre-existing cell to other cells, and they all happen 10 times more frequently than spontaneous mutation. All right, you know about plasmids, transposons. That's it for chapter 8. Any question? Well, I'm actually on time. Let's move to chapter 1. Oops, let me get back there. Oh, crud, what am I getting out of here? Ah, advanced sharing option. There we go, share. Get out of here. Chapter 1, the microbiological world in you, and a little bit of chapter 10, classification of microorganisms. Know the terms. Mainly, let's go over the brief history of microbiology, and then know the importance of microbes in the ecosystem on the planet Earth, and the importance of them to humans, like we use microbes to make different products for us. And that's talking about the importance of microbes in the nitrogen cycle, where all the steps of the nitrogen cycle with the red arrow are happening because of a microbe. Obviously, you should know that microbes are important in making rock and making oxygen, important in sewage treatment, important in bioremediation. Should know about the naming and classifying of microorganisms. The naming began by Carolus Linnaeus, and he simply invented a two-name system using Latin names to name everything. That system was still so good, we're still using it today. He did begin the classifying of organisms, but he classified everything into a plant or an animal. And as you know, we have more organisms than just plants and animals. And so his classification system is not used, but he's the first one to begin both the scientific names and then the scientific classification. Any question about any of that? So you should know that all life on Earth can be classified into the three-domain system. The domains are defined. We know which organism belongs to which domain by sequencing the DNA of the organism. And if we were to get a new organism, find a new organism on the planet Earth, we could simply sequence the DNA, and the sequence would be either much closer to the bacteria, so it belonged to the domain bacteria, or to the archaea, so it belonged to the domain archaea, or it could be most closely related to the eukarya, in which case it would belong to the eukaryotes. So the domains are classified on the sequence of their DNA. And all organisms in the domain bacteria are more similar in their sequence of DNA to all other organisms in the domain bacteria than to any organism outside the domain bacteria, meaning it'd be much more closely related to the sequence of the DNA to bacteria than a domain such as archaea. We're talking about a organism like, let's say, E. coli. It would be much more similar to other bacteria in the domain bacteria than to any organism in archaea or in the eukaryotes. You can then classify the domain into kingdoms, but scientists disagree for how many kingdoms there are and what the classification scheme is for classifying them, meaning the kingdoms are undefined. So there's arguments about what organism belongs in which kingdom. And there's also an argument about how many different kingdoms there are. Some people use a three-kingdom approach, and that's simply making a domain a kingdom. So that would be simple. Each domain is also a kingdom. There's the five-kingdom approach. There's the six-kingdom approach, and that's the approach that we use. It's the last approach that was used in biology. The trouble was that fell apart when we sequenced the DNA. Now we know there are more than six kingdoms, and I guess we always knew that because the kingdom Protista was not a monophyletic group. It was simply a eukarya that was not a plant, not an animal, and not a fungi. So this was always a group that we understood had multiple origins, meaning multiple groups in it. And then there's the 9-kingdom approach, the 15-kingdom approach, the 20-kingdom approach, and then the higher order, 30 on up to 50-kingdom approach. We're only going to discuss the 6-kingdom approach, where there are two domains and two kingdoms, bacteria and archaea, and each domain is a kingdom for these two. And then domain Eukarya, which has 4 kingdoms: Kingdom Plantae, Kingdom Animalia, Kingdom Fungi, and Kingdom Protista. You should know about the cell types, meaning prokaryotes and eukaryotes. All right, let's go on to the brief history of microbiology, which largely was concerned over the debate of whether spontaneous generation of life could occur. Could, especially a microbe, spontaneously generate? And that was a large part of the early history of microbiology. This is the only date you need to know. About 3.5 billion years ago, we have the first fossil record for life in the rocks. And that is, we have fossil records to the ancestors of bacteria. In the history of things, you should just know what the event was and who did it. Like Janssen developed the first compound microscope. Surprisingly, the microscope was never used to look at any organisms until Robert Hooke, in what is that, like 75 years later, used a microscope to look at plants, and he discovered little boxes that he called cells. And that marked the beginning of the cell theory, that all living things are made of cells. Or as you know about in the history of microbiology, there were 2 hypotheses. One, that microbes could spontaneously generate, and that others, microbes could only come from pre-existing microbes, and that's known as the theory of biogenesis. The first evidence against the spontaneous generation of life came from Francesco Redi's experiments, and he simply had jars with a wire mesh over it and then meat inside the jars. And if you left the jar open, maggots would appear in the meat, but if you had a wire mesh over it, the jar, there were no maggots in the meat. And Francesco Redi's experiment was important because it's the first one disproving spontaneous generation, at least of complicated life forms. And it's also the first documented controlled scientific experiment. So this is the first time somebody wrote up a controlled scientific experiment. We'll come back to Antony van Leeuwenhoek, but let's go on to Louis Pasteur disproved spontaneous generation by having the swan neck flask, putting the media in a flask with a very large neck, heating the neck, bending it into a swan neck, and then heating the media, sterilizing it, and then leaving it, and the media never got microbes. And that's because the microbes would be unable to come down from the air. They'd fall here, and gravity would not allow the microbes to come up and then contaminate the media. And so Pasteur's experiment disproved the theory of spontaneous generation by microbes. Now, before that, let's go back. There's an important person called Antony van Leeuwenhoek. He was the first person to see microbes, where he was such a good microscope builder and lens grinder that he was able to see microbes wherever he looked. We'll just word it that way. He found microbes wherever he looked. And there's a microscope of his that's still alive today, and it's probably in a museum. And he was able to see cells so small they were not seen by other scientists for almost a year. not a year, 100 years, 100 years, meaning a century. And he was actually able to see bacteria, as shown here. All right, so you should know some of these other events, like Koch developed the streaking for colony isolation on a solid media. Pasteur before that came up with the idea of aseptic technique. And then you should know a little bit about the germ theory of disease. And that was the idea that microbes could cause disease. It was actually Dr. Koch who showed the first microbe, Bacillus anthracis, that causes a disease. Oh, you should probably know Edward Jenner, the first person to come up with a vaccine. Alexander Fleming, the first person to discover an antibiotic. He discovered penicillin G, which interestingly was not the first antibiotic used in the clinic. It was actually sulfonamide, the first antibiotic ever used in the clinic, one of the sulfur drugs. The normal microbiota we already talked about, and then emerging infectious diseases. Any question about chapter 1? All right, I'm not going to talk much about the other chapters because there's only a few questions on each of those chapters, but you should at least go through the objectives for those chapters in your studying. For chapter 2, I think there's only one question on chapter 2. Any question about any of that? If not, that's it. And I'll say goodbye if there's no questions.
Thank you.
All right. Thank you.
Good night.
Good night.
Thank you.
Thank you.